All right, welcome everyone. We are looking forward to UCLA's presentation today for the Undiagnosed Diseases Network Grand Rounds. My name is Emily Glanton. I'm a genetic counselor and the Associate Director of the Data Management Coordinating Center at Harvard Medical School. I'll go through just a few basics about the UDN for anyone joining us for the first time or in a while, and then I'll turn it over to our presenter for today. So Yvette, can you go to the next slide? Thank you. So we have a research network of clinical sites, research cores, the Data Management Coordinating Center, and our sequencing core all across the US. And there's more information about all of our different sites and groups on our website as well. The next slide, the UDN's goals are to improve the way that we diagnose and care for patients with undiagnosed diseases, facilitate research into the causes of these conditions, and to create an integrated and collaborative research community so that we can figure out the best ways to serve these patients. Next slide, please. So every quarter we publish a quarterly report that's available on our website at the link there on the About Us page. And from the July quarterly report, these are the updated numbers. So about 30% of our participants have been diagnosed, and we've evaluated about 520 variants by the Model Organism Screening Center, completed RNA sequencing for about 780 participants. And this graph is just showing a little bit more detailed breakdown of some of the sequencing of the approximately 2,400 sequences that have been done in the UDN. And you can see the breakdown by the type, exome or genome, or sometimes both for some of our participants, and then by age, pediatric or adult. And in green, you can see the rate of diagnostics. Thanks, Eva. Next slide. So just to remind you about our grand rounds for today, we welcome you to ask your questions in the Q&A text box, and we'll answer those questions at the end of our presentation today. And the information today is presented for educational and informational purposes and can be used for personal and non-commercial uses if the content and references remain unchanged, and no part of the presentation can be published without prior permission from us. And then there is an hour of CME credit for today's presentation. So for anyone wishing to claim CME credit, you'll just go to that QR code or this link here and sign into the Stanford CME account to create your account and complete the evaluation to claim that credit. I will also have this slide up at the end of our session today, so you'll see that again before we finish. All right. And with that, I am really thrilled to turn it over to Yvette Corona, who is a bioinformatician at the David Geffen School of Medicine at UCLA, one of our UDN clinical sites. And without further ado, I'll turn it over to you. Thank you.
Okay, so thanks for having me here in the UDN Grand Rounds. Today I will present what we have learned in this project of using long-read sequencing for diagnosis of rare diseases. So I'm a bioinformatics scientist in the Nelson Lab at UCLA. So start with the definition of rare diseases. In the US, it's defined as any disease or condition that affects less than 200,000 people, and that is about 1 in 1,659. In Brazil, there's a different definition that accounts for like 1 in 1,500, but the most common definition is 1 in 2,000 people. So Russia and Peru have different definitions, and there are some countries that actually do not have a formal definition. I think it is very important to have this because it's tied to having policies related to rare diseases. Then rare diseases are actually, I like to have the sentence that rare diseases are common, and that stresses the idea that we should have people that put effort and resources into the study of rare diseases. So we have more than 7,000 rare diseases identified. There are 25 to 30 million Americans living with a rare disease. That is around 8% of people in the US. But in the world, there's like around 350 million people, and that is like around 4.4%. And I think I was just getting these percentages preparing for the presentation. And I think the difference in percentages that you see here might be due to precisely the diagnosis of rare diseases. Also, rare diseases can affect all ages. As you were seeing in the slides from Emily, we do have cases that are pediatric cases and other cases that are adult onset. I actually joined the UDN around in 2022, and I was not in this area of rare diseases and human genetics. So you sometimes think of a genetic disease or that somebody is born and then it's onset in infancy. But we have diseases that are adult onset and actually that you're an older adult when you get these diseases. And it is specifically important for this talk because when you have adult onset cases, maybe you don't have parents that are available to do the genetic analysis, and it is difficult to diagnose these cases or identify the cause when you don't have the parents. So having a trio whole genome sequencing, it's different. It's more challenging. It's better having the trio than not having the trio and having only the proband. Then there are, in the world of rare diseases, there are two main challenges that I can list here. So the diagnosis is one. It takes more than 5 years to get a correct diagnosis. And around 50% of people are diagnosed, but the other 50% don't receive a diagnosis. And the other challenge is the treatment. So there are more than 90% of rare diseases that are still without an FDA-approved treatment. But in the UDN and in this talk, we're going to just focus on diagnosis. And this slide, I took it from a colleague, Dr. Huang, that tries to show that there are different technologies that can help us diagnose rare diseases. So this was specifically done for neurodevelopmental diseases. So you have chromosomal microarray, clinical exome sequencing, and reanalysis. You have also whole genome sequencing that now it's more available to do it clinically. And in research, we used on a routine basis whole genome sequencing and transcriptome sequencing together. So all of this have, as I mentioned, a diagnosis rate of 50%. But the other part of cases that are not diagnosed or solved, we believe, we are in what we do is to try to identify new technologies that can help us identify identify the cause of other rare diseases. So I have to say from this slide, I think it's very important to know that reanalysis it's also by itself improves the diagnostic rate. And that is because every day there's new diseases that are discovered worldwide. And then if you have an exome or a genome that was negative 5 years ago with new annotations, then doing reanalysis by itself, it can actually improve, diagnose somebody. Then continuing with this conversation, so the current technologies have a diagnosis rate of 50%. So what is done clinically is all of that I mentioned before, including transcriptome sequencing, but most of the time is targeted transcriptome sequencing. So maybe you already have a variant that you identify through whole genome sequencing, but it's predicted to alter the splicing, for example, and then you do targeted transcriptome sequencing for that specific gene. But in research, we do whole genome sequencing with transcriptome sequencing for all the genes. And then we have new technologies that are being tested and developed. So one of them, one of the ones that are listed here, I think it was important to note. So deep sequencing is something that we have that's available since a long time, but some discoveries or recent discoveries point to low variants that are mosaic but have mosaicism for 2 to 3% that are gain of function, that can actually be important for to the development of some diseases. Then what we have done also in our lab is to have different ways of using transcriptome sequencing, including these methods that I list here, including nonsense-mediated decay inhibition, ultra-deep sequencing, or enhancement of low-abundant transcripts. And for today's talk, the focus is on the use of this technology, which is long-read sequencing at the level of genome and transcriptome. So this slide shows more or less how short-read sequencing is done. So you have the genome, and then you use you fragment the DNA to be able to sequence it, and then retrieve those or sequence those reads that can be from like 50 base pairs 300 to 200, 300 base pairs, and then align them to a reference genome. That's what we usually do. So the premise is that a 50 base per long fragment or a 300 base per fragment of DNA is unique, and that you can align it to a reference genome in a specific position. So that premise breaks when you have repetitive regions, and I'm not even talking at this point to any genetic variation that is going to be pathogenic. Just we have in the genome regions that are repetitive regions, and regions that are actually that are known to be problematic for short read. And then you add the part of having something, some genetic variation that can make it even more complex, so copy number duplication or complex structural variants that basically break the premise that a DNA sequence of 300 base pair is unique, and then you are actually not able to align and resolve some regions in the genome of a patient, of any human. So long read sequencing in practice. So what we did then, this is the protocol we have. I mean, the steps are very simple. Then we have a sample, prepare it, and therefore sequencing. So all of this was done by Dr. Nassef in our lab. And then we actually receive raw data and then process and analyze the data, right? And it was done this in part by George in our group. And then we have, these are all the samples that we sent for sequencing for this project. We have a total of 21 different data sets from 17 samples. And it was 19 from these 21 data sets. This was 19 DNA sequencing from 16 different participants of 14 affected participants in total. So some of them, most of them actually have short read DNA. So we're able to compare the variant calls from short versus long. And then we also had two of with RNA sequencing. In this presentation, I'm not going to cover RNA sequencing. We're still working on understanding more how we can use this genome-wide. So what we did is that for these two cases, we had specific questions to ask that we wanted to use long read for, but we are not using yet this data like genome-wide for analysis. In average, we have in the Hi-Fi dataset, so we're actually using PacBio. So I forgot to mention that for long read, I mean, there's two big technologies or two companies that are doing long read. So one is PacBio, the one that we use, and the other one is Oxford Nanopore, ONT. But we're using PacBio. And so in average, we got 8.1 million reads, and the read length is 11,000 base pairs. So compared to 300, so 300 it was using short read, and then right now we have reads that are expanding like 11,000 base pairs. So our yield, it was not in average 91 billion base pairs, and our coverage is in average 30, like close to 30x. I mean, this is an improvement of this specific technology from PacBio. I think we have our lab, and I was not involved in that project at that time. I was not yet in the UDN. So I believe that they had lower yield, lower coverage for our past project using this technology. So right now, I mean, it's an improvement, and I think this is actually very good for what we do, diagnostic of rare diseases. We use two different cores. So the one in UCLA, the UCLA Neuroscience Genomics core, has one of this. Provides the services of sequencing long read with PacBio, but also UCSDG core. And then something I wanted to mention, because one of my objectives today is to actually give you like what it was our hands-on experience. So to because we're building our data set, I mean, we have 21 samples, and then the idea is to keep building on increasing this data set that we have. There has to be some way of annotating everything and then keeping track of what we have. So we're actually using the data dictionary of the Gregor Consortium, and I encourage everybody that is going to start a project like this to keep track of all all these metrics. And Gregor has as a group of scientists that have thought about what are the key metrics that you have to keep track of. So it's actually available in GitHub. So you can go to GitHub to the Gregor Consortium page of GitHub, and then they have the data dictionary there. So once we actually merge the data, our data with others data set, I mean, it's already readily available to have all these metrics and compare them if they're a good quality, all of them, or similar quality. Then this is the set of tools that we used. George implemented a pipeline that it was provided by, or it was a pipeline that PacBio provided, or it was provided by us in their GitHub. So this is the set of tools. So it was alignment by PBMM2, and then the variant for variant calling, PBSV for structural variant calling. It has phasing as well, a tool for tandem repeats, and then other tools that are annotated here. And first, right now, I'm going to present two cases that we used long read to solve these cases. So the first case is an 11-year-old female with a history of suspected neurodegenerative disorder with progressive loss of motor and speech, as well as cognitive and memory abilities associated with worsening seizures and hypertonicity. So this is, it was onset at 2 years old, and the EEG demonstrated some seizure activity. And so a few months following the onset of seizures, then movement and stability began regressing, and stability began regressing. So it's a case where this girl developed very severe dystonia and contractures, and then genetic and metabolic disorder workups have been negative. This is the pedigree for this case. So where we have now 13 years old in when this pedigree was done. And then you can see that it had two other sisters that were healthy. And then on the mom's side also, it was nothing was identified as related to this girl's disease. And then on the dad's side, and actually I think dad was not available for genome. This was a dual whole genome sequencing, but it had a similar presentation or some overlap of the phenotype. But in the patient, in the Provence case, it was onset at 2 years old. And in the dad, the phenotype that overlap, it was onset at 38. And even the paternal grandfather had some similar phenotype onset at age 50. Then we had prior genetic testing, before they joined the UDN. So they did dual whole exome sequencing that was negative, whole exome sequencing reanalysis that also was negative. So microarray panel that it was negative, and ataxia panel from Athena Diagnosis that was negative as well. Then the application was submitted in November of 2019, and I think everybody knows what there might be a gap here. So we have the COVID-19 pandemic in 2020. That after that happened, then we submitted it for dual whole genome sequencing, and then there was a clinical evaluation as well. And after we received the raw data from the dual whole exome sequencing, we did an in-house analysis that identified an HTT repeat expansion. So this analysis was done by Li Kai Wang, and we used Illumina Expansion Hunter. That I know that this figure is it looks really, really tiny dot here, but this is the original image that Li Kai used to then identify that there was a repeat expansion in HTT of what we thought that it was 124 repeats. So after that, we sent the DNA for different analysis, and NAGME was able to give me the reports of all this analysis. So in 2021, then we received from Baylor, we received the dual whole genome sequencing report that was negative. Of March of the next year, we also sent to Baylor a specific analysis for Huntington's disease, and it was negative. And then in May of that year, 2022, then we sent to Prevention Genetics, Huntington disease via HTT CAG repeat expansion analysis, and also was negative. So from our analysis, we were pretty sure that there was something going on there, but for some reason it could have been, it was not able to detect using this analysis. So we thought about that this case was a good case for to do long read. And in May, we did proban only hi-fi sequencing. And then also the next month, we received a report from this other lab that also identify a repeat expansion. So first, I'm going to show you what the negative results and the methodology of those results, just to have an idea of what I think that it could be, they were negative. So in the first one, which is the whole genome sequencing duo, so it's 150 base per parent reads. And in the methodology, they list Illumina Dragon to do variant calling and copy number analysis, and they use Illumina Manta for structural variant caller. They do not list what we used was Expansion Hunter, so they don't list a tool to do repeat expansion. So I'm not sure, because it is the same raw data that we're using to do this analysis. So I'm not sure if they do this, they use this tool specifically to identify repeat expansions. And then this other test is they use a restriction enzyme. We think that it could be a problem, that maybe it has some rare variant that are interfering with the analysis, maybe with this restriction enzyme. Something that I have to note is that in the results, they only list one allele. And it's mentioned here in the methodology that it says additional PCR analysis using different primer sets will be performed for samples with a single allele in order to rule out allele dropout. So I'm not sure if it was done or if it was only the original set of primers and the restriction enzyme to do this analysis, but this is the report for this analysis. Then prevention genetics, so identify homozygous for two normal HTT alleles. And then they said in the methodology and then the limitations that they actually have identified cases with 180 repeats. So these tests could have potentially identified something that it was really large, as the case of our patient, but they know that they have limitation, that they might carry an allele that doesn't amplify, and then they cannot see it. And then LabCorp, which is the test. I mean, this is the test that they actually identified something that it was large and abnormal. So they do a test that it was developed by Medical Neurogenetics. So it is, they identify a heterozygous repeat expansion. So the second allele, they only listed at larger than 100. So they use something called triple repeat prime polymerase chain reaction, TPPCR. And they say that, false negative results are rare, and that the assay itself doesn't allow anything larger than 100 repeats. So anything that shows larger than that for them will only be reported as more than 100 repeats, but they don't have the exact number. So using long read, we were able to observe that there was something abnormal. And then this is the previous figure that I showed you, that it was saying that it was 126. So in this genome browser view, I wanted to show you what is the whole genome sequencing data looks like compared to the HiFi. So in the whole genome sequencing data, the mother looks normal. And in the proband, then we can see that there's something different going on here. And you can see it from these reads that look misaligned, but what is happening is that they're actually kind of like insertions, or is actually the repeat expansion. And that's why our expansion Hunter analysis showed repeat expansion. And in the HiFi data, and this is more like clean. This is cleaner data, and I really like the HiFi data, how it looks like. So everything is homogeneous. Then we have a heterozygous, so everything that has this gap of 12, so that is going to be the normal allele. And then everything else, you are going to have either something that looks here like an insertion. So here actually says 820. So this is actually another one that says 795, so close to 100. And then other reads that are going to have this kind of mismatch base pairs. And that is also going to be part of the repeat expansion. And then the analysis that we did, it was alignment followed by tandem repeats. So it's a program called TRGT, and this is a visualization tool that they have. And it is very clear here. So what the call was that it was 264 repeat expansion. And this is, I think, one of the longest that has been reported. And as the world knows, or the scientific community knows about Huntington disease, is that the length of the expansion is negatively correlated with the age of onset. So we believe that because this repeat expansion was so long, that's why it was onset really early, like at 2 years old, it was already showing some signs of the disease. Then I'm gonna go to the second case. This is a 6-year-old male with severe keratosis pilaris milia with congenital hypotrichosis universalis. He was born with no hair, no eyebrows, no eyelashes. He has this diffuse milia on cheeks, mid-face, upper and lower extremities, all that since birth. And it was initially thought to be ectodermal dysplasia, but it didn't develop abnormal teeth or nails. So then it kind of changed what they think the condition was. So the family history was nothing related to, so it doesn't have family history of that, and consanguinity was denied. And there was some testing prior to the UDN. So he had done a 75 gene panel for ectodermal dysplasia that was negative. Also, a chromosomal microarray that was normal, and exome sequencing that identified 3 VUSs, but not related with the patient's phenotype. And then the application was submitted in November of 2023, and a little bit less than a year after, then we had proband-only HiFi sequencing for this case. And then this is what we identify. It's a 20 kb duplication in XQ26.1. So maybe this is a little bit hard to observe, but this genome browser view is both HiFi, but the top one is the case that I'm presenting, and then the bottom one is it's a control case. So we have here, I hope that you can observe that it has this duplication or a structural variation here, and it's actually this 20 kb duplication, and it's flanked by these two genes. So this is actually an intergenic region that, to be honest, like we normally don't look there. It was thanks to the referring physician that knew about this publication that it was done. I actually don't have the date of this publication, but she thought that the referring physician was almost sure that this was the cause. because it was very, like the clinical description, it was very similar to what they're described here. And they have a range of duplication in this region, and we found it. I mean, it was very like directly going to this region, and then we found this. I mean, we didn't know if it was going to be. I mean, I like when we were doing the analysis, we had the long read to do like a genome-wide analysis, but we went directly here and saw it. And it's a duplication that yeah, it was very clear. So in the publication, they mentioned that even though that is closer to the gene in the right, it actually affects expression on this gene, ARHGAP36. Okay. So something else that we've learned, we long read, I mean, something that in the premise that we have regions that are not being able to be resolved with short read. So here I'm going to show an example of these problematic clinical genes. So this is one of the genes. So I think it was yeah, like last week that a group of the UDN UCLA clinical site. So they were telling me about a lot of genes that are actually problematic and that they're hard to resolve using short read. I mean, I have a list like maybe around 10 genes, and there could be more. And I'm going to show just two that are, because it's very clear. So you have SMN1, which is the gene that I'm showing here in the bottom. So it goes from so this is 5' and this is 3' end. And in NOMAD, the coverage in the genomes, it's really poor. So this track here, so it should go from all this way. You only have coverage on this gene at the 3' of the gene. And then so you have these green areas that are covered, not in all the genomes that NOMAD has. But you also have this other track that I hope you can see. I mean, you don't have to look at the detail, just look at that. This shows the variants that are annotated in NOMAD. So these variants fall directly to the regions that were covered by the genome. But all the other rest of the gene, it's not covered by the genomes in NOMAD, and also you don't have information of variants. So now what I'm showing you here in these other tracks is a set of, I think it'll maybe like 15 or 14 samples that we did with HiFi that I'm able to cover better than that NOMAD's genomes. And you can see that there's a lot of variation, and it's going to be a common variation so a lot of these variants are actually common, and these are unrelated individuals. Therefore, a lot of these variants, my idea is that are going to be common. So this highlights so the idea that a lot of common variation is not annotated in NOMAD. Therefore, when we do long read sequencing, we're actually going to also do variant calling, and a lot of the calls are going to be common variants that are just missing in NOMAD. Therefore, we need to develop more or develop or sequence other individuals using the same technology to be able to identify what are the variants that are actually common now using this new technology. So this is another gene that is a problematic gene. It's from the same family, SMN2, that you can see also that it is covered only on the 3' end of the genome. Also has variants annotated in NOMAD, mostly on the 3' end of the gene. And you also have genomic variation that I'm suspected that is going to be common throughout the gene and is not annotated in NOMAD. And talking in general, what we found is that we have more, like in terms of small variants, we have more small variants called by HiFi and than by whole genome sequencing. The overlap is really large, so that's good because we do want them to overlap. But we have more calls done that are unique to HiFi. We have also a set of calls that are unique to whole genome sequencing, and I don't know what these are, so I only did a correlation of these variants related to to quality, so the QC quality metric. And so they do have lower quality compared that the ones that overlap, but there's a set that are also high quality. So I think it's important to understand why we have variants that are missing. And then the set of variants that are unique for Hi-Fi. So some are going to be the ones that are not annotated in NOMAD because simply the technology of short read couldn't call them and couldn't actually resolve those regions. And structure for in the area of structural variants with Hi-Fi. So as I mentioned, we have this pipeline that for structural variants it uses PBSV. It is available in GitHub. And then what we also saw, so one of the problems we identified when we were trying to do analysis of the structural variants is that there's a large number of structural variants now called by PacBio. So the problem with that is that to do analysis, so a lot of part of that step is manual. So we have to kind of narrow down to a set of variants that are possibly pathogenic, and we use that by overlapping with common variants, as I was mentioning. That's why we use NOMAD. But in the area of structural variants, it was just so many more variants that we're actually not able to narrow it down to a human to be able to for a human to actually do the analysis of all of the variants that are not annotated in NOMAD, for example, or that could be pathogenic. So what I wanted to show you is that I think there's a need still to develop more tools to prioritize these variants, but also the need to develop or to have more controls to filter out variants that are going to be common. Then here we have some tools that we were testing and using. So PBSV is the one that I mentioned. The last release was on this year on February 26th. And then the PacBio also developed Sawfish. And I just checked and they actually had a release on June 18th of this year. And there's another tool that is not developed by PacBio. It's independent. So it's called Needler. And the thing about Needler is that they actually are working together with a group that are sequencing, or the plan is to sequence from the 2000 genome projects, 10,000 genome projects, or 1,000 genome projects. They want to sequence more. So we have here actually used this tool when they had 150 genomes already sequenced with ONT. So now they mentioned, I just checked, and they have 450 controls. So that is going to help tremendously. And what I'm also going to mention here, that even though that they use ONT and we're using PacBio, so it is also long read. So they're also going to identify structural variants that were not identified with short read. So we can use those controls sequenced by ONT to then filter down things that are going to be more important. So for example, we had for this, it was just one example, one of the samples that we had. PBSV identify in the order of 50,000 structural variants, Sawfish in the order of like 35,000, and Needler annotated 22,000. And I have to note that it doesn't annotate translocations. But from 22,000, using the controls that they had, we were able to narrow it down to 1,200 unique, so that they are actually not present in the other controls. So this is of really high importance to have more controls to be able to then do analysis on the set that are either unique or that are predicted to be pathogenic or to affect function. Then there's another tool that I want to talk about, which is Hi-Fi SV Topo. That is a visualization tool. And this is an example of what it can do. So this is an example of a very simple tandem duplication and with whole genome sequencing. So this is a trio. And then we have the Hi-Fi in the bottom. So in the whole genome sequencing, if I see just the whole genome, so this is the data of the mother, father, and proband. And if I just had the whole genome, I would say that this region was problematic. So I couldn't actually make sense of, I mean, maybe there were other tools that would help me identify what was going on in this region. But if I was not particularly interested in this gene and I was just doing like a genome-wide analysis, this region wouldn't tell me anything. And actually, I'm not saying that this was a solution for this case. So I'm just saying that I couldn't resolve it and I would just ignore it, right? And then in terms of hi-fi, so this is a hi-fi data, the last track, and this is very easy to see that it was what was happening. So this is a tandem duplication, and what SVTOPO does is actually tell me what is the topology precisely of what is going on. So the reference is ABCD, and then what the variation is ABBCD. So it's a tandem duplication in this region. And then this is another example of how we use SVTOPO. Just this is an inversion. We have the same sample, mother, father, and proband, where we have a structural variant inherited from the mother. What we see in the whole genome sequencing is actually reads that I think this coloring is telling me that it could be a translocation. But what is really happening is an inversion. And then SVTOPO was able to help me then understand what this messy, or what looks just messy, it was actually an inversion. So the reference is ABC, and then the alternate is ABC, but B is actually inverted. And then in summary, I mean, we have used this technology to diagnose rare diseases patients. We know that it uncovers new types of variants, so we're going to have more variants than before. But then we actually need more controls to then filter them out, and then have a set of pathogenic variants that we should focus on for each patient. And then we know that it actually helps in phasing. I didn't show you here, but it's going to be in particular important in cases where we might have like a compound heterozygous variant, but we don't have the parents, and then we can do phasing for this for that case. So what I also tried to tell you is that bioinformatic tools are still maturing, but that doesn't mean that you shouldn't do long read. I mean, we need more control. So if you have the opportunity to do long read, you should join the effort of getting them sequenced. So we have more controls, and we're actually able to solve cases or solve more cases using this technology. And then that's basically what I have prepared. I just want to acknowledge everybody that contribute to this work. So the California Center for Rare Diseases, and then the UDN UCLA clinical site. The PIs for our clinical site is Dr. Nelson and Dr. Martinez. We have then people involved very directly to this project is Dr. Nassif and George. And I think I'm ready to then to go to the question section.
Thank you so much, Yvette. It was great to hear those case examples and walking us through some of the things that you all have learned, a lot of trial and error. So thank you for sharing your wisdom with us. We have a few questions so far, so I'll go ahead and start with those, and everyone attending can feel free to add some questions to the Q&A as well. So the first question is if you can speak to the differences between ONT and hi-fi in terms of the repeat expansion analysis.
Oh, I cannot, like, I actually haven't used ONT, so I'm not sure what are the tools that are available for ONT. So like I can show you the one case that I presented where we did repeat expansion, which is this one. And it was very, very easy to identify this case, to resolve this case. So with these tools, but I'm not completely sure about how would I do it with ONT. I'm sorry.
No, that's okay. Thank you. It's good to know. All right. So the next question is if you can comment on any software that you are using for SNV or copy number variant interpretation.
Yes, so let me show you the pipeline. Yeah, so this is the pipeline that we're using. And then with the PBSB, we get the variant calls, and it's basically a VCF, and then we load it to we use VS Clinical, which is the VARSIC from Golden Helix, and try to use that for filtering. However, as I mentioned, so using our normal pipeline that we would use with short read, it's just retrieving thousands of variants that are supposedly, I mean, that couldn't been filtered out. So we are still left with thousands of variants to check manually. So we haven't been able to then kind of do that, like narrow down basically let me go now to structural variant here, kind of to narrow down these thousands of variants of structural variants that are retrieved or called to narrow it down to something that is possible for us to do analysis. So we have tools, but I don't think they are actually right now at the level of helping us solve cases yet.
Great, thank you. And a question about the SV topo. So was SV topo better than IGV for visualization using the same sample of PacBio data?
Yes. So what IGV, which is this view, would give you is the reads and how they're mapped. And then you have a view of how the reads kind of piled up. But as you can see, I mean, it gives you this information of, it gives you some information of what is happening in this region. But SV Topo gives you like a representation of the topology of what is happening. So from just looking at this, which is just IGV, I cannot tell you what is actually happening. I mean, I have the annotation or the call from the PBSB, which is the equivalent of MANTA, and it might be telling me that it's a duplication, but I actually in IGV by itself, like I cannot tell you if it's a tandem duplication, if it's what is happening. So SV Topo was very useful for helping us really understand what was happening in the region. Yeah.
That's wonderful. Thank you. All right. So there's some interest in the second case. So there's a few follow-up questions there. So one question is there any follow-up still being done for that second case, for example, to see if that intergenic duplication causes transcriptional or altered protein levels?
So there's not in our from our group. However, believe this is the paper that makes the reference or they already did some tests, and I think what is happening is that it's affecting the gene that is in the right, which is ARH-GAP36. So we are actually not planning on testing that in our patient. So what we are reporting is just that it has this duplication and it's already reported to be pathogenic.
Great, thank you. And the next question also about this case is kind of out of curiosity for the diagnostic accuracy of the referring provider who had that instinct about the diagnosis. So just curious about what kind of provider that was and if they were a geneticist or what their background was.
Yeah, it's a geneticist from UCLA, and yeah, like I was already also like shocked, like because I mean, I wrote down, like she shared with us what her thoughts were and wrote it down, but I actually haven't encountered a case where the referring physician actually knows what the answer is and you just have us checked it, right? I mean, for me, it was, oh, I have to do a genome-wide analysis. But first, let me check here. And it was actually the solution. So it's very, I mean, it just shows how important it is that communication with the referring physicians. And we actually, like, as analysis, like, read every detail, everything that the information that they give us, so we can focus our analysis on that. If I didn't listen as an analysis, if I didn't listen to that, and I just started with a genome-wide analysis, I actually would have missed this because my analysis right now, it's very hard for me to solve these regions that are intergenic. And I'm not sure if this is actually annotated right now in ClinVar as pathogenic. So I would have missed it. So I'm glad that we didn't ignore that.
Yes, that's a very good point. The importance of collaboration between our clinicians and our bioinformaticians.
Yeah.
All right. And then I'm gonna keep going on this one case that some folks have some questions on. Is this a de novo duplication? Do you know?
We don't, we believe it is. We believe it is. I don't have the genome of the mom. I don't remember what they're going to do about it. We just believe that it is a de novo duplication. And all the cases that have been reported, they have been de novo. But I don't have the genome of the mom. Oh, of
Okay.
the parents, I'm sorry, yeah.
All right. Thank you. Oh, yeah, you do say it's proband only there. Okay. And
Mhm.
then a little bit, zooming out a little bit about this technology in general. So, do you foresee this technology one day being a replacement for exome or genome and microarray, or a way to get, both types of data in one test? Or do you think that this will continue to be a last stop reserved for the cases that earlier testing is not successful for?
So how I see the evolution of that, I think it's similar to this. So I believe that exome is still used in the clinic, even though that I'm sure genome covers more. So I think that is gonna happen. I mean, I'm hoping that long read at some point, it shows. I mean, we already know that it's gonna be useful. But I think it's gonna be basically kind of like another resort after maybe it has been done whole genome sequencing. Maybe I'm wrong. Like I'm not, like I have never done predictions on biotechnology, but it can replace, I think, the other ones. But I mean, when did we have whole genome and it's still whole genome and we still have whole exome happening, right? So I think it's a long road for that to happen.
Yes, that's a great point. All right. And then also related to kind of in general recommendations. So you've talked about the importance of sequencing additional controls. And this person is wondering if you have any recommendations in the choice of samples, given that most cases are genome negative.
Set of samples that can be used. I think you need to do. Yes, my recommendation is that you select maybe a group of cases, potentially. I'm going to show you what we did. It was five, no, three groups of cases. So one, it was like muscular diseases. The other one was like micro. Um, what is it called when they're born with microcephaly? And there was microcephaly, and then I don't remember the third kind of group of cases. And then we have around like five samples for each group of diseases. And then I think the idea is that you have to be sure that it is a genetic disease, right? I mean, you have to be sure that you're looking and that there's a genetic cause for the disease and it is basically hidden somewhere, right? So, and prior to send it for sequencing, I highly recommend to do a whole genome sequencing reanalysis to make sure that it's negative because we actually encountered one case that we sent. And then while I was analyzing the long read, I was also doing reanalysis of the short read, and I identified the cause by the reanalysis itself. So I recommend everybody to do reanalysis, like a fresh reanalysis prior to send it for sequencing.
Very good guidance. Thank you. So a few more questions related to the tools that you're using. So the first one is, what tool are you using for the tertiary analysis after variant calling?
Yes, so we have different tools, but the main one is we use Golden Helix VS, the VS Clinical. I think it's the original one is called Varsig, but we use the clinical version, and that's what we mainly use.
Thank you. And then another question that someone might have missed, but did you also use the colors DB PacBio data to filter for allele frequency?
I did not. So what is it called? Colors Pack Bio?
Yes, and it looks like the O's are lowercase, so it might be an acronym. Yeah.
Okay, I know. I didn't know of it. I'm gonna look it up. Another thing that I forgot to mention that PacBio data is actually five base reads. So it also has information of methylation, of DNA methylation, and that's something that we haven't explored yet. So it's something that can be useful for understanding if we have a variant of how if it's affecting or not. But that's a set of data that we have, and we still haven't been able to analyze it.
Great. And I've shared in the answered questions that this Shruti had shared the PacBio. It's a consortium of long read data. So you should be able to see that link in the chat as well. Okay, we have one last question, at least for now, and then I think we'll just move on to wrapping up with the CME code. So the last question is a case example. So recently at one of our other clinical sites, we had a case that was strongly clinically consistent with Shwachman-Diamond syndrome, and genome was negative for the gene associated with that condition. And according to the reading, it seems like there is a highly analogous pseudogene. So they were wondering if you've ever run into this type of situation with a pseudogene, and if you think that long read might be necessary to detect the homology.
So that's what I heard about this, like what I call them problematic genes. That here. So that's what the clinicians in the room were telling me. So I'm not a geneticist, and I do the analysis of this, and basically every new case for me is I don't know about the diseases itself, but this is what they were telling me, that there are a set of genes that because they have pseudogenes, they're problematic, or like regions that are not very easy to be resolved by short read. So this is my idea of trying to talk about this, that long read will be able or can resolve this. So actually, if they know the gene, I actually can check in my data set if I'm able to, if I can cover, like if in my set of samples, if that gene was covered or not. So before they send it for sequencing, I can do that as well.
Okay, that's great. So yeah, if the person that asked that question wants to send us an email afterwards, I can put you in touch. Thank you. All right, so let's go ahead and move on to those closing slides. And we have the CME code at the very end that I want to make sure people have access to. So we also have a few upcoming grand rounds. So our next one is in October. And then we have the following quarterly series in 2026. The registration links are on our website as well. And if you have any questions about the UDN in general, you can contact our help desk at the information there. And then the last slide is just the CME code again. So you can go to that QR code or link to claim your CME for today's session. And again, I want to thank Yvette, you and the UCLA team so much for today's great presentation and all of your hard work on these cases. So thank you.
Thank you, and thanks everyone.